So it's my pleasure then to welcome next Professor Nevan Krogan from UCSF. He's a director for the Quantitative Bioscience Institute, and I'm sure he will follow up on much of the same topics as Adolfo was talking about. So please go ahead, Nevan. Thank you so much for being here.
Hello. I assume I'm okay. So it's very pleasure to be here. My friend to speak after my other friend, Adolfo Garcia Sastre, and I'm going to give you a update on some ongoing work here on SARS-CoV-2 that we've been involved with over the last year at UCSF and with the Quantitative Biosciences Institute, which is where I work. And all the work I'm going to be discussing is under the umbrella of the QCRG, the QBI Coronavirus Research Group. This is a group of labs that have come together to work on COVID-19 over the past year, involving hundreds of different scientists here at QBI. And we've actually broken the QCRG up into multiple different subgroups, 12 different subgroups, and I'm going to be speaking more about one of the subgroups that's focused on structural biology in just a few minutes. And I should also say that this has been a great worldwide collaboration as well, with a number of different academic institutions, including Mount Sinai, especially with Adolfo here, and also industry connections. We've been working with a number of different industry partners as well. And to me, this is one of the great silver linings of this pandemic, is to see how fast we can all move when we work together. And the challenge here is to try to keep this infrastructure and spirit in place so that we're better prepared for the next pandemic. And then the larger question is, why aren't we working on all diseases like this? And the hope is we will be in the future. And this is a point I'll touch upon at the end of my talk here this morning. And then the other drug I want to spend a little bit more time on is another translational inhibitor, Aplidin. It inhibits a translational elongation protein called EIF1A. There's the structure of Aplidin in the top left-hand corner. It's actually from the company PharmaMar. It's being used to treat multiple myeloma. It's called Aplidin when it's used to treat cancer, and plitidepsin when it's used to treat COVID-19. There it is, the structure of it binding to its target, EIF1A. This wasn't work from us. This was from a different group. And interestingly, it comes from this sea squirt that lives exclusively to Spain. And all the data I'm going to show you has really come from Adolfo and Chris White in a paper that was recently published. So very excitingly, it's very potent plitidepsin or Aplidin in human cell lines. This is their work in HEC293 cells. It has a sub-nanomolar IC90 value. And in this particular cell system, it's 30 times more potent than remdesivir. That's on the left. On the right there, I'm showing you it's very potent in primary lung cells as well, again, at a much lower concentration than remdesivir. And Adolfo has this great mouse model to look to see what effects drugs have after you can infect mice with SARS-CoV-2. What you do here is use an adenovirus to express human ACE2 in mice so that you can infect the mice with SARS-CoV-2. You then add drugs, and temporally you're taking out the lungs and looking at the viral titers. And on the right there, you're seeing about a 500-fold decrease of infectivity with Aplidin at a much lower concentration than is used for remdesivir. So it looks incredibly potent. And Adolfo and I have been interacting with the company PharmaMar based out of Spain, and they've been conducting clinical trials on COVID-19 using plitidepsin, and they concluded a clinical trial here in the fall that looks very promising. We're working with them to put out a paper on this particular clinical trial, and based on all of this, very excitingly, Aplidin has actually entered a phase 3 clinical trial for COVID treatment at 27 different sites across 12 different countries. So the hope here is that we'll get some good results here on this phase 3 clinical trial, and this will be another weapon against the fight of COVID-19. And I think it's particularly interesting because it actually these connections I'm alluding to came from this proteomic analysis that we did with Adolfo and Pedro. And just a few comments here about the variants, which of course now we and many others are studying. And one of the values of targeting the hosts as you start to tackle the variants. So when you target a host protein as an antiviral, you don't have to worry so much about resistance because we don't mutate as fast as the virus. And I would argue that the virus would never mutate enough to overcome its reliance on a particular host protein. So the logic here is that if you're able to inhibit the older lineage of SARS-CoV-2, you should be able to get these new variants. And indeed, Aplidin or plitidepsin is just as potent against the UK variant as we see with the older variant. And we see the same thing with the Brazilian variant and the South African variant. And this is done collaboratively, again, with Adolfo and Greg Towers and Claire Jolly at the University College London. So I think there's great value here in targeting a host protein, because if you can get SARS-CoV-2 and SARS-CoV-1, the logic also would be, hopefully it would be applicable for SARS-CoV-3. And of course, we all know that the next coronavirus is coming, hopefully not soon, but it'll come eventually as well. And I think I'd be happy to take questions at the end of this session.
So maybe I can go on with the second question now to Nevan. So again, we have a lot of questions, but so there is a question from one of the audience asking if you plan to expand this protein-protein interaction work to other coronavirus respiratory viruses, and if this would help to potentially find pan-viral therapeutics. I think in part you have already answered this question for the SARS-CoV, but I think he wants to know if you are extending this.
Yeah, actually, before the pandemic, we were working with a program called Fluomics that also alluded to it, at UBs, where we were doing similar things with influenza, so flu A and flu B. And right now we're also looking at other respiratory viruses like RSV and other coronaviruses too, like ones like OC43, ones that aren't so problematic. And just as you point out, one of the goals is to look across all of this data and find commonalities, and there are commonalities, very exciting commonalities. And then one of the bigger goals is to exploit these commonalities to come up, hopefully, with pan-viral therapeutic strategies. So there's some great, cool biology that's overlapping, and then the challenge is working with people like Adolfo. Can we translate those biological commonalities to therapeutic value? And we're working hard on that right now. And actually, that's one of the things I'm most excited about as we generate more and more data.
I found your talk really interesting. And as a virologist and a microbiologist, we've spent all our lives trying to find unique patterns or unique features of microbial proteins so we could target them specifically. And now your approach is totally the opposite, which is fantastic, which is targeting those proteins. And as you said, they're not prone to variants and mutations from the microorganisms. But the reason why we do that, targeting specific microbial functions, is to minimize cellular toxicity. So how do you see this? Do you see this as a problem in your approach?
I guess a few things I would say along those lines. If you think about drugs for all their diseases, they're targeting human proteins, and it's been successful, obviously, in the other context. But the other thing to point out here is that using plitidepsin as an example, it's being used actually in these COVID-19 trials that we're working with Pharma at much lower concentrations, and it's being used to treat cancer. But then also think about it, infection is acute, whereas these cancers are chronic often, right? And you sometimes take these drugs for weeks, months, and even years, but when you're treating an infection, maybe you got to take it for 5, 6, 7 days. So taking these things into account, I don't think toxicity will be such a problem, although you have to be wary of it. It won't be as problematic as, say, some of these drugs being used to treat these other diseases.
Yeah, I agree with you. Thank you.